Hello everyone, my name is Kumar. I'm a PhD student working under the supervision of Professor Kumar Johri at Jawaharlal Nehru University, New Delhi, India. Today I'm going to present my work on sulfate transporter from a root endophytic fungus Serendipita indica. Sulfur is one of the essential elements required by all living organisms, including plants. Sulfur is a constituent of proteinaceous amino acids such as methionine and cysteine, and some of other biomolecules, including glutathione, vitamins like biotin, thiamine, chlorophyll, coenzyme A, etc. Sulfur is also involved in disulfide bond formation in proteins. Sulfur is also a component of several secondary metabolites of plants and is required for plants' physiological function, growth, and development. The sulfur demand in plants is dependent on the type of species and stages of development. Most of the sulfur in soil is present in organic matter and hence not accessible to the plant. An ionic form of sulfur, that is sulfate, is primary source of sulfur for plants that are generally present in minimal amounts in soil. It is water soluble, so readily leaches out of the soil. So sulfur is very important component of plant metabolism, and its analysis with different dimension is highly required to improve the overall well-being of plant. The deficiency of sulfur lead to stunted growth of plant and ultimately loss of yield. However, an association with a symbiotic fungus can greatly improve nutrition uptake by the host plant. In a recent study by our group, identify and functionally characterize a high affinity sulfate transporter from root endophytic fungus Serendipita indica. SISLT fulfill all the criteria expected of a functional sulfate transporter responding to sulfur limitation. SISLT expression was induced when S indica was grown under low sulfate conditions. SISLT is a 763 amino acid long with a predicted molecular weight of 83.2 kilodalton. It is a member of SLC26 family. The SLC26 family of transporter possesses a membrane domain comprised of 14 transmembrane helices as shown in figure B. The overall topology of this domain resembles a pseudo two-fold symmetry, with each half containing 7 transmembrane helices oriented in opposite direction within the membrane, resulting in an intertwined structure formed by 2 inverted repeats. SLC26 transporter possesses a C-terminal stas domain directly linked to transmembrane helices number 14 by a short linker, as we can see in this figure number B. To gain more quantitative understanding of sulfate uptake by SISLT heterologously expressed in yeast, then they conducted uptake assay with radio labeled S35. We first grew the wild type yeast strain BY4742 and HK14 mutant, transformed with either empty vector or with SISLT in sulfate replete condition before transferring them into sulfur starvation medium for 2 days. We then measured the sulfur uptake by adding 10 micromolar S35 labeled Na2SO4 for 4 minutes. Both BY4742 and HK14 transformed with SISLT showed a strong accumulation of radioactive signal, with 296 picomole sulfate for BY4742 and 146 picomole for HK14 transformed with SISLT. In contrast, the HK14 mutant transformed with empty vector took up negligible amount of sulfur with a very low radioactivity count of 0.2 picomole sulfur equivalents, as we can see easily in figure number 2A. These data suggest that SISLT has the potential of supplement the yeast HK14 mutant. We then probe the uptake kinetics of SUL1 and SUL2 in BY4742 and HK14 transformed with SiSLT under a range of 35 labeled sulfate concentration from 1 to 100 micromolar. Sulfate uptake in both strains um, followed a typical Michael-Menten kinetics with a apparent Km of 15 around 15 micromolar and Vmax value of around 1.92 picomole per minute per OD of OD at 650 for HK14 transformed with SiSLT and a Km of 8.2 micromolar and Vmax of 3.2 picomole per minute per OD of 654 BY4742. SiSLT was expressed in yeast and purified using different chromatography techniques. Figure A shows the chromatogram of SiSLT purification. Peak P1 represent the SiSLT fraction. Fraction also loaded on SDS page to check the purity and presence of purified protein, and we got a sharp band around 75 kDa, which confirms the presence of pure SiSLT. Purified protein was then used to make negative stain grids, and grids were observed under T12 electron microscope. Data was collected at 30,000 x magnification, and data was processed using CryoSpark software. 2D classification was done and we got different classes. Average of SiSLT represents wide distribution of SiSLT particle in different orientation. Particle size found around 20 nanometer. I would like to thank UGC Government of India for providing me fellowship, IUBMB for providing Wood Allen Fellowship, UCSF and JNU for providing facilities and instrument for conducting the research, Professor Atul Kumar Johri, Professor Robert M. Stroud and Dr. Nitesh Khandelwal for helping me throughout the project. Thank you.